A white shark and a Greenland shark both fit inside one familiar word, but that label hides radically different lives. One is a fast regional endotherm. The other can live for centuries in cold water. Treating them as interchangeable predators erases the differences conservation and ecology actually need. Today, we will audit 4 layers: evolutionary history, body temperature, lifespan, and ecological role, without pretending every shark shares one operating system. The first surprise is not that sharks are ancient, it is how much unique history sits inside individual branches of their family tree. Shark ancestors extend beyond 400 million years in the fossil record, but living sharks are not unchanged fossils. A 2018 conservation study assembled a taxon-complete tree covering 1,192 sharks, rays, and chimeras. Its molecular backbone contains 610 species, with taxonomic constraints used to place the rest. Across that tree, the average species embodied about 26 million years of unique evolutionary history. That number is an average, not a claim that every pair of sharks split at the same date. The useful lesson is unevenness. Losing one distinctive branch can erase far more history than losing another. Heat branches are only the start, because some sharks even manage body heat differently. Most sharks are ectothermic, while a smaller set can keep selected tissues warmer than the surrounding water. This is regional endothermy, not a mammal-like promise that every part of the body stays at one temperature. The distinction matters because the measured advantage was speed, not a universal license to live everywhere. To understand this thermal split, we must examine the unique circulatory adaptations of the Lambdidae family. Species such as white sharks and short-fin makos use counter-current heat exchangers that retain heat produced by active muscles. The familiar anatomical term is rete mirabile, closely arranged vessels that exchange heat before it is lost to cooler blood. Narrative tracking and performance data linked regional endothermy with faster, sustained swimming. That finding supports a performance advantage, not a claim that every warm-bodied shark reacts faster in every encounter. Many other sharks remain ectothermic and function within very different activity budgets. So the thermal payoff is precise. Some sharks warm selected tissues and swim faster on average. The study did not measure a fixed extra prey requirement for every endothermic shark. It also found no broader horizontal or vertical thermal distribution for the endothermic fishes in the comparison. Warm tissue changed performance more clearly than it changed the temperatures those animals occupied. Now compare that speed result with a life history record measured by an entirely different tool. Inland sharks move through cold northern waters, but the landmark study tested age, not a complete theory of slow metabolism. Their longevity question required a clock that could persist inside a cartilaginous fish. Researchers used proteins formed in the nucleus of the eye lens because that material remains metabolically inert after development. Radiocarbon values in those lens nuclei offered age estimates with unusually wide uncertainty. That uncertainty is part of the result, not a footnote to be removed for a cleaner story. To uncover the true scale of this slow-motion existence, scientists turned to an unexpected dating method. Conventional hard-part growth counts were not available in the same way they are for many bony fishes. The team therefore radiocarbon-dated eye lens nuclei from 28 female Greenland sharks. Their analysis supported a lifespan of at least 272 years. The largest animal had a central estimate of 392 years, plus or minus 120. That range is why a precise birthday or historical anniversary would be false precision. The secure payoff is still extraordinary. The data supports centuries of vertebrate life. The paper did not compare every reef shark lifespan or document a universal age of senile decline. It did estimate sexual maturity near 150 years, revealing how slowly replacement can occur. That single life history fact already shows why shark management cannot assume one recovery speed. But the differences between these groups run even deeper than their lifespans and body temperatures. Inside a white shark genome, researchers found selection signals associated with genome stability and wound healing pathways. The important word is associated, because a genome scan is not a clinical superpower test. Popular claims often turn those associations into the myth that sharks are immune to cancer or disease. The study sequenced and analyzed the white shark genome, one species inside a much larger radiation. It reported evolutionary signals in pathways relevant to DNA damage response, repair, and wound healing. Those enrichments are biologically interesting, but they do not equal perfect genomic stability. They identify candidates for further study, rather than proving what every gene does in a living shark. The paper did not demonstrate instant wound healing or the prevention of every mutation. It also cannot make one white shark genome uniform across approximately 520 described shark species. Other lineages require their own genomic and physiological measurements. So the molecular armor payoff is a boundary. Promising repair-associated signals are not cancer immunity. Nothing in this genome paper supports taking shark cartilage as a cancer treatment. Turning evolutionary associations into a medical product claim would go far beyond the evidence. The honest result is compelling enough without adding invulnerability. A different comparative record shows diversity on the outside of the head, electroreceptive pores. Elasmobranch electroreceptors help with functions including prey detection and orientation, while their distribution varies among lineages. Kempster and colleagues linked receptor number and pattern with phylogeny, head shape, and ecology. This is a comparative map, not one universal shark sensor layout. The receptors terminate in jelly-filled canals commonly called ampullae of Lorenzini. Odd or elongated heads can spread sampling pores across a different geometry than compact heads. That geometry can change how electric information is sampled around the animal. But the source does not support a rule that every deep-sea shark has fewer pores and compensates with enormous eyes. The sensory payoff is variation tied to anatomy and ecology, not a ladder from primitive to advanced. Taxonomists still group modern sharks within a real evolutionary framework. Diversity does not mean false kinship. They are cartilaginous fishes, though their skeletons include mineralized tissues rather than being soft cartilage throughout. Shared features help define the group, while species-level bodies and life histories remain highly varied. A shared label is therefore taxonomically useful and ecologically incomplete. Paleontology also warns that many extinct shark-like fishes fall outside the crown group of modern sharks and rays. The mistake is not grouping sharks, it is assuming the group performs one ecological job. That assumption creates the final trap, calling every shark an apex predator. Predator effects depend on species, prey, habitat, and whether mortality or fear changes behavior. The trophic payoff begins with a limit. The review does not say every shark drives a top-down cascade. For marine top predators that do exert strong effects, prey can respond both by being eaten and by changing where or when they feed. Those risk effects can propagate through a community even without an immediate kill, but context decides whether that mechanism is strong, weak, or absent. Evolutionary distinctness adds another conservation lens. Some threatened branches contain unusually large amounts of unique history. That is what the phylogeny measures, not a claim that shark lineages are as distant as mammals and birds. It also does not substitute for species-level data on abundance, movement, reproduction, or metabolism. The Greenland shark illustrates why life history belongs beside evolutionary distinctness. Its eye lens evidence supports the longest known lifespan among vertebrates. Estimated maturity near 150 years makes population replacement exceptionally slow. The oldest estimate still carries a plus or minus 120-year uncertainty. At another extreme, regional endothermy raises sustained swimming speed in some active sharks. The same comparison found no corresponding expansion of their occupied thermal range. Either result lets us infer the physiology of every bottom-dwelling or poorly studied species. Conservation therefore needs species data plus evolutionary and ecological context, not one generic shark stereotype. One name can contain a fast, warm tissue hunter, a centuries-old cold water survivor, and hundreds of other solutions to ocean life. The category stays real. The stereotype is what fails when physiology, lifespan, and ecological context enter the frame. Shark policy built only around the largest famous predators can miss small, deep water or unusually distinctive branches. A policy built only around one food web model can also assume risk effects where they have never been measured. The better sequence is species identity first, then life history, habitat, threat, and demonstrated ecological role. For Greenland sharks, late maturity immediately changes the recovery question even before every ecosystem interaction is known. For regionally endothermic sharks, higher sustained speed describes performance without predicting a broader thermal home. For white sharks, genome associations identify research targets without conferring a medical miracle on the entire group. One word can organize a family tree, but only evidence at the right scale can explain what an animal actually does. Subscribe to Oddly Evident for more animal labels that become stranger and more useful when the evidence splits them apart.